to hear next week. So come for both. Dr. Abid received her MD from Dow Medical College in Karachi, Pakistan. She then came here to the U.S. For her pediatrics residency training at the University of Medicine and Dentistry in New Jersey. And then fortunately for us, she then came over here to complete her child neurology fellowship, fell in love with us, but spent 5 years over at the University of Oklahoma and took the first chance she could to come on back here. She's now one of our neuromuscular specialists, actually has a strong clinical and research interest in muscular dystrophies, but it sounds like she has also now branched out into the world of Friedreich's ataxia. So please join me in welcoming Dr. Abid.
Thank you, Tuan, for a kind introduction. And as you can see, I'm going to talk about Friedreich's ataxia, which is a fascinating disease with very interesting pathophysiology and has diversity in the clinical presentation. So I'm going to present a few of my cases also to show you the variability in the phenotype. This is the CME talk, so I have to have this slide, but I have nothing to disclose. These are my objectives of the talk, so by the end of this talk, everybody will be able to describe the basic pathophysiology and genetic aspect of FRDA, recognize the clinical feature, and list new emerging therapy of this condition. So Friedreich's ataxia is a pretty old disease. It was first described in mid-1800 by a German professor of medicine, Dr. Nicholas Friedrich, and he described as a disease characterized clinically as ataxia and pathologically by a degenerative atrophy of the posterior columns of the spinal cord that could affect several children of unaffected parents. The way he described the localization and also the possible genetic aspect of the disease in the era where there was no MRI and the genetic test available is pretty impressive. So as per Dr. Nicholas Friedrichs, that parents are not affected, and later it was confirmed that Friedreich's ataxia is a autosomal recessive condition. So to have this disease, a patient should be homozygous for the mutant gene. So you can see in this picture that if the parents are carrier, with each pregnancy, there is a 25% chance that the child will have the disease, 50% chance that the child will be carrier, and 25% chance that the child will not have any problem. There is no de novo mutation has been described in this condition except a recent paper just published this year, which shows that there is one child with the maternal transmission and one spontaneous mutation. So Friedreich's ataxia is a very common hereditary ataxia. It comprises more than 75% of all hereditary ataxia before 25 years of age. It's very prevalent in Europe, Middle East, South Asia, and North Africa, but not documented in Native Americans. The incidence is 1:50,000 in Caucasian, but in Central Europe it is a little high, it's 1:20,000. The carrier prevalence is 1:110, and both male and female are affected equally. So where's the problem in Friedreich's ataxia? It is due to the mutation in frataxin gene, which is present on chromosome 9, and penetrance is complete. This is one of the trinucleotide repeat disease, which is due to expansion in the non-coding sequence. There are other conditions like trinucleotide repeat disease, which have expansion in the non-coding sequence. One of them is myotonic dystrophy. And there are some conditions which have expansion in the coding sequence, like exon, and some of them are Huntington disease or spinal cerebellar ataxia. So 96 to 98% of the patients have homozygous, they are homozygous for guanine, adenine, triplet repeat expansion in intron 1, which is a non-coding sequence. And repeat can vary from 66 to 1700, but the most common repeat in disease-causing repeat is 600 to 1200. Normal people have repeat from 7 to 33, and some people may have repeat from 34 to 65, which is called premutation, and it can expand in intergeneration transmission. And expansion can be possible in both maternal and paternal transmission. 2 to 4% of the patients are compound heterozygous, so they have one allele of trinucleotide expansion, and the other allele could be either point mutation or deletion. Less than 1% of patients with further phenotype, they have no GA repeat expansion. So it means that they don't have Friedreich's ataxia. You have to have one GA repeat to have a diagnosis. So if they have phenotype looks like Friedreich's ataxia, then you have to think about other conditions like ataxia with the ataxia with the oculomotor apraxia, or spinocerebellar ataxia with axonal neuropathy, or other conditions like ataxia telangiectasia. In adults, it could be multisystem atrophy with cerebellar type. Frataxin gene has 5 exons, and you can see here in normal state, normal GA repeat, you have normal production of frataxin. When there is expansion in the intron 1, usually the expansion caused changes in the configuration of DNA, so it becomes sticky and it cannot be transcribed. That's why it caused decreased production of frataxin. Frataxin is a mitochondrial protein, but is encoded by nuclear DNA, and it is present in the matrix of mitochondria. It expressed in all cells, but in variable level. It depends on the mitochondrial content of the cell. It's most abundant in heart and spinal cord, and followed by liver, skeletal muscle, and pancreas. And these are the systems which are involved in Friedreich's ataxia. So what frataxin does, it has multiple functions. The main function is, number one, iron metabolism. So it acts as a carrier or also donor for iron for synthesis of multiple enzymes. For example, ferrochelate, which is one of the enzymes required for synthesis of heme, and also iron-sulfur cluster. It also helped transport of iron back to cytosols. So iron enter into mitochondria and then used for different function and then go back to cytosol. And cytosol iron is very important because it actually increases the expression of frataxin. So if you have decreased cytosol or iron level, it can cause decreased expression of frataxin and make the condition more worse. Second function is iron-sulfur synthesis, which is actually prosthetic group for respiratory chain enzyme, including aconitase and complex 1 to 3, and it also helps for DNA repair. The third function is energy metabolism, which is through the iron-sulfur cluster, and it leads to synthesis of ATP and antioxidants like superoxide dismutase or glutathione. And these antioxidants actually reduce the oxygen radical, which are toxics, which produced during the energy metabolism. So if you have absence of antioxidants, or basically the main pathophysiology in Friedreich's ataxia is increased oxidative stress, which can cause damage to the protein, lipids, and mitochondria, and cause cell degeneration and apoptosis. So in this diagram, you can see the same function of frataxin. Is it encoded in nuclear DNA, and then from cytosol it enters in the mitochondria. Iron enters in the mitochondria, and then both together helps to have iron-sulfur cluster, which is a prosthetic group for the respiratory chain enzyme and cause production of ATP. And also frataxin directly activates superoxide dismutase, which is one of the antioxidants. And there are production of oxygen radicals through the energy metabolism, and also if the iron starts accumulating in mitochondria, it can also increase the level of oxygen radicals through phantom reaction. It's the same diagram to show the function, and due to lack of frataxin, the function which are affected is basically this enzyme. You can see ferrochelate, which is involved in the synthesis of heme. Then you can see iron here, which is cause increased production of oxygen radical in the absence of frataxin, and even the iron can cause phantom reaction and more iron radicals in mitochondria. And the lack of iron sulfur cluster decrease ATP production, decrease antioxidant production. And again, this shows the function of iron sulfur cluster. It is present in both cytosol and mitochondria. And you can see it involved in tricyclic acid cycle. It also is also in the electron chain transport system, and also other functions, and which is related to Friedreich's ataxia, but it also causes some other reactions, synthesis of different enzymes and metabolism pathway. This diagram to just to show the normal mitochondria, and then in Friedreich's ataxia there is accumulation of iron, which is toxic to the cell, and it causes increased production of oxygen radical through the phantom reaction, and that leads to the damage of lipid protein and mitochondria and cause apoptosis and cell regeneration. So what are the areas involved in central nervous system in Friedreich's ataxia? So basically it involves the posterior column, spinocerebellar tract, cerebellum, which gives you both sensory and cerebellar ataxia. It also causes loss of sensory cells of the dorsal root ganglion, loss of peripheral sensory and motor neurons that leads to axonal neuropathy, and also it involves pyramidal tract. So the constellation of symptom in Friedreich's ataxia due to involvement of cerebellum, sensory pathways, and corticospinal pathway. Before the era of discovery of the gene and the genetic test, Friedreich's ataxia used to be diagnosed by diagnostic criteria. The first criteria was proposed in 1976 by Quebec Cooperative Study in Friedreich's ataxia. And according to these criteria, that patient have to have symptoms before 20 years of age, progressive ataxia, lower limb reflexia, decreased vibration, weakness, and dysarthria. And then Harding and Fila actually modified these criteria a little bit. Like Harding has only added sensory neuropathy, and then Fila added cardiomyopathy. After the discovery of gene and the genetic testing, 25% of the patients were not fulfilling the criteria proposed in this list. So we stopped using this criteria for diagnosis. And FORTA can be classified as possible, probable, definitive, but nowadays we just use definitive diagnosis based on the genetic testing, because all patients are diagnosed by genetic testing, so it's all definitive. But if you don't, if you see a patient and you're suspecting, you can still put probable or possible. So clinical manifestation varies with the size of GA repeat. So larger the size of GA repeat, early the onset of disease, early confinement to wheelchair, rapid progression, early involvement of heart. The mean age of onset is usually around 10 to 15 years, but it can be as early as 2 years and as late as 6 years. Other presentation could be late onset Friedreich's ataxia. These patients usually have a smaller repeat, like less than 500, at least in one allele. And maybe they have compound heterozygous, or maybe they have more non-GA repeat interrupting the GA repeat. The onset is usually after 25 years of age, and they usually present with mild symptoms and a slow progression and late confinement to wheelchair. Very late onset Friedreich's ataxia usually start after 40 years, and again they have a small repeat, less than 300 in at least one allele, and maybe compound heterozygous. And their symptoms mimic like multisystem atrophy, cerebellar type in adults. The classic presentation of Friedreich's ataxia is gaze instability and progressive ataxia. And they also have absent ankle and knee jerk, Babinski sign, dysarthria. Dysarthria is usually scanning and slow jerky speech, absent vibration and proprioception sense, which also leads to sensory ataxia. Weakness initially proximal and later involved distally. Mixed axonal neuropathy usually cause atrophy of the small muscle in the feet and leads to foot deformity, which is very common in Friedreich's ataxia, and also cause hamartosis pascavis. these patients also have other system involvement. So 25% of the patients can have optic atrophy. Visual symptoms usually seen in 20%, but if you do a testing like optical coherent tomography or visual evoked response, then we will see abnormality, even mild, in almost 100% of the patient. Oculomotor abnormality include square